All right, quick review. Quick review that we know what DNA is made of now. Deoxyribose, phosphate, the ever-changing nitrogenous base, of which there are 4. We talked about how Watson and Crick and Rosalind Franklin kind of unwillingly figured out that double helix structure. The double helix being the idea that there's 2 separate strands of DNA spun around each other, held together in the middle by those weak hydrogen bonds, and that there's a sort of a head and a tail end to each strand. And so that it only works if they're opposite directions. And the word they used for that was anti-parallel. Those strands zipped together in the middle by those weak hydrogen bonds. That middle part really makes up the code. And the way those bases pair with each other via those weak hydrogen bonds, it only works in 2 configurations. Adenine only makes those bonds with thymine. Guanine only makes those bonds with cytosine. So you're always going to be able to tell what's on the other side. If you know one strand of DNA, you know the other. And I told you how I remember it, which is via the shapes of the letters, very kindergarten-y. By the way, you're going to have lots of cutting today, also very kindergarten-y. A's and T's have straight lines, C's and G's have curvy lines. Any questions about that stuff so far? So we use a word for the way those bases fit together. That word is complementary. We call them complementary base pairs. As in A is complementary to T and G is complementary to C. Just a quick English note that complementary with an E in that spot I've circled is a different word with a different meaning than complementary with an I in that spot. Do you know the difference? What each one means? Like if I say, go ahead.
The complementary angles.
That's the right one. That's the one we're using here. Why are they complementary angles?
Because when you add the 2 angles together, they connect with 3 angles.
They go together perfectly to form a right angle, right? A right angle. Yeah, they go together. That's the complementary with E part. Like if I say, Ben,
yes,
really like your sweatshirt.
Thank you.
Looks really good on you.
Thank you.
That's me being complimentary with an I to Ben. I'm paying him a compliment, right? But if I say, Ben, that mask and your sweatshirt, they go together perfectly. They're complementary.
Yeah.
That's the E one. It means they go together, right? Okay. I don't want to freak out. So now, this is kind of the training wheels section. We're going to go through a process that's kind of the easier of the processes that you're going to learn. We're not going to do yet how DNA codes for protein. That's the big idea of the chapter, okay? How DNA actually makes you the way you are. What we'll do first that teaches you some of the basic concepts is how does DNA make more DNA? How does DNA replication work? Well, first of all, just to access some prior knowledge here, when does DNA replication happen specifically? Good. S phase of the cell cycle, specifically the parts of the cell cycle when mitosis isn't happening, which we collectively call interphase. Good. So S of interphase is when this happens. Got to do it before you're going to do either mitosis or meiosis, because you're going to need a double set of DNA to divide a cell. And here's where we're going to do some drawing. This is kind of a diagram-heavy chapter. Got to be able to visualize stuff. I will be giving you animations, except all mine are currently broken. I didn't realize that all my DNA stuff uses Flash, and I can't get Flash to work. So what happens first in DNA replication is we got to get at the code. If we're going to copy the code, we got to be able to access it. And to access it, we got to break those weak hydrogen bonds. So there's an enzyme you make called DNA helicase. I think I've told you before that in the names of enzymes, whatever comes before the ACE is usually the thing that that enzyme breaks down. So helicase breaks down the helix, right? It splits the helix down the middle. To give us a simplified view of that, let's draw a simplified, very simplified molecule of DNA and show the helicase running along in between the bases and separating them. So now we can access the code. And the thing that your cell turns out to be really good at is matching up complementary bases to a single strand of nucleotides. You have enzymes called polymerases that are very good and very fast at sucking in all the nucleotides that are floating around free and sticking them on in sequence to single strands that are exposed. A lot of this is about shape, right? The A's are the right shape to form hydrogen bonds with the T's. The C's are the right shape, not like the kindergarten thing I was telling you, but like real life, that they have the right kind of crags and points and stuff to form those hydrogen bonds. And so that's how your cell knows, is those are the right shape. And it puts the right things in the right sequence to match up with the exposed strands that are here. But the problem here is, even though those enzymes are really good at that, you're going to need to do it separately on both strands. You're going to need an enzyme to do it on this strand and an enzyme to do it on this strand. And they only work in one direction. In other words, a polymerase enzyme, which you'll write down in a minute, can only run along a strand of DNA in one direction. And since the two strands are anti-parallel, they're going to have to go opposite ways. So, first of all, the name of the enzyme now is called DNA polymerase. DNA polymerase has to be shown where to bind to the DNA by a small stretch of RNA called a primer. That's the green thing here. So DNA polymerase won't bind unless there's a little primer there to show it where to bind. So you stick the primer on there. But the polymerases have to go in opposite directions, adding bases as they go. Remember, the thing that's paving the way for this to happen is the DNA helicase, which is running along the double strand, separating, breaking the weak hydrogen bond. Any questions at this point? So far, very simple. Right? We got something that split the strands, and now we got two things that are adding complementary bases to the strands. But it gets complicated now just a little bit because of the fact that the helicase is running in one direction. The helicase, think of your 6 feet of DNA. Think of the helicase starting over here and separating the DNA as it goes. If the helicase is going this way, this polymerase has no problem. The polymerase that's going the same direction as it, fine, it's just following it. It's just adding bases as it goes, trailing the helicase as it unzips. But think about the one that's going the opposite direction and having to add DNA this way. You know, it started here where the primer is. As soon as this thing opened up, primer went here, DNA polymerase went here and had to stop because this is where the helicase started. So now the helicase is still going. What you're going to have to do is lay down another primer, stick on another polymerase, and backfill. And keep backfilling and backfilling and backfilling because this polymerase has to go the opposite direction as the helicase. It doesn't totally make sense yet. Let me show you this picture. Maybe that'll help a little bit. Now I know there's a lot of details here, but here's your DNA, your original DNA together, right? Here's the direction that the helicase, which is the little blue triangle, helicase is unzipping in this direction. Well, this polymerase that gets to go the same direction is in luck. It gets to keep going and going and going behind the helicase. The one that has to go the wrong way has to keep going and keep going and keep going. It needs a primer every time. By the time this is done, another enzyme is going to have to run along and smooth out where all the primers are and fix that strand of DNA. Does that make sense at all? You there? So, okay, in words, what I just said, helicase can only go one direction. The polymerase that gets to go that direction behind it, that's called the leading strand. The strand where the polymerase gets to go the same direction is called the leading strand. The other strand, the strand where the polymerase has to go the opposite direction of the DNA polymerase, or sorry, of the helicase, that's called the lagging strand. The scientist who discovered that you have to keep backfilling on the lagging strands, his last name was Okazaki. So we actually call all the little backfilled pieces separated by primers that you have to do on the lagging strand, we call those Okazaki fragments, which I like to say. Okay, ask questions if you got them, please, because if you've got them, somebody else does too. We'll all benefit from it. Do you get it? Do you understand the concept of Okazaki fragments? It's the only real hard part of this. It's the only challenge. Otherwise, DNA replication is fairly straightforward. It's just that directionality of the strands that gets in the way of doing this, right? You can unzip the thing, and the polymerase that gets to go the same direction has it easy, whereas the one that has to go the wrong way, you have to keep adding new polymerases and splicing them together. Okay, I trust you. What we wind up with after this is all done is two identical double strands, right? We've copied the DNA from one double strand into two identical double strands. How new are they? How new is each identical double strand? Don't overthink it. How new? Like this and this. How much of that? How much of each of those things is new DNA? Half. These are half new double strands, right? We've used each half of the old strand to build new halves. So half old, half new. And that's the last thing. That's the thing I'll leave you with here is we have two identical DNA molecules, each one half old, half new. Yes, this is the last drawing of the day. As I said, I will find animations that work. Mine are all suddenly broken. The last thing is there's no way you could do this in a reasonable amount of time with your entire 6 feet of DNA using just one helicase, starting at one end of the 6 feet and unzipping all the way to the other end. It's not how it works. That would take forever. So what you do instead is you make thousands of helicases and thousands of polymerases, and you do this in thousands of places at once along the 6 feet, and they all kind of meet up together. Which means you can get S phase done in hours rather than months. All right, rather than make you think a whole lot more, we're going to have some kindergarten time now. You're going to be cutting out some paper shapes for yourself. These are the models we'll use to walk ourselves through replication and the rest of it. Today, you are responsible only for the top 2 pages. I will be giving you a gallon-sized Ziploc bag to store your little paper shapes in. You can also fold up and store the uncut stuff so far. And these are things you'll be expected to have in class for the next week or so. All right, so you have the rest of the time to cut. We're using your scissors. If you didn't bring any, I do have a tub. You should have those top 2 pages, which are identical, cut out for tomorrow.